happy to give the floor to Silke van Wijngaard on both specificity and interactions.
Okay.
And I'll let you take it from here.
Yes, thank you, Alena. Thank you, everybody. Also, people online. Hello. Yeah, I think this is going to be a bit of a different talk, but that's nice, and I think that's very cool. And I think there will be a lot of connections to make. And this is going to be much less clear and than Alena's talk. So be ready for just diving into things, and it can be a bit chaotic. So that's my thing of ready to go down the rabbit hole, because when Detmer asked us, okay, it was host specificity, and like, host specificity. Actually, do I really research on host specificity? No, I do look at interactions, diversity, and then when you touch upon it, but when you think about it, oh, let's see. So let's go.
I'm sorry, guys, if I did assignments that were a bit.
So I want, as I said, I can quickly introduce myself for people that don't know me. So I'm currently in Finland at the University of Turku, but of course I am not Finnish. I come from here, from Belgium, and I started actually my scientific career, like PhD in Switzerland at EAWA. On the supervision of heat on heat rate. So that's when I will really kind of encountered for the first time these organisms. Did my PhD on that, and of course after the PhD, nobody wants to continue right away, or like it's like so stressful period. You want to do something else, and I quickly did that, changing my mind and going just to Uruguay, looking at parasites of Daphnia, but then quickly this also had some stopover in Belgium again with the ParAqua-COST, but I quickly kind of like realized, no, I want to work on that. This is what I really love. And luckily, I found the group of Hans-Peter Brossa at IGV. So he was kind of at that time point very interested in aquatic fungi. And so he was immediately enthusiastic about also working on these hydrates, but he actually at that time point did not really have somebody working on that topic. So I spent really quite some time there as a postdoc. And also spent a very short time in Austria, where also Serena has been working with Martin Kainz and Robert Tachnik. And then I kind of like, so all you see this kind of things like my background is in freshwater, but then it's kind of started to point that come out like, okay, these hybrids, they are actually like, are they only freshwater? No, we also kind of start to encounter them in marine systems, but the knowledge was kind of much less present there. And that kind of dragged me like, okay, let's see, like, well, I want to fill a bit of that kind of knowledge gap too. I realized that actually in the Baltic Sea, which is a quite big ecosystem, and very well characterized in terms of phytoplankton ecology, there was basically like nothing really going on, or like nothing there about hydrates. And that's when I kind of decided, okay, I will write this project grant, and luckily I got funded, and I'm there now since in 2021, where I have a small group, aquatic microbial interaction group, with one PhD student, Jonna, and one master student at the moment, Siru, we are doing now research in the Baltic Sea on hydrate interactions. Also have a little bit of a private team and a side project. So if you go on the website, we have a little bit of a simulation modeling. Starting with, I'm not a theoretical biologist, but I kind of like try or like I really kind of like this agent-based modeling simulation approaches, where you can kind of with traits and you can combine, you can create a virtual lab. So that's kind of my idea of to use this kind of virtual lab and going to look at the things, the black boxes actually that we don't have any empirical data yet, or if we have, we can kind of use that. But this is a little bit, I'm not going to talk about that now, and it's a bit on the side. We are waiting for funding also to kind of continue that. And Kiki is also waiting here because he's the debugger of that program, so very important. But now coming to the talk, so that was very in a nutshell me. So let's go and talk about host specificity. So how is it defined, like commonly? And I have taken this from Pola, which is, I think, quite a famous scientist, like working on this aspect a lot with like, in macro host systems. And we can kind of like very simply say it as the extent to which parasites can exploit different host species or genotypes. So, and why is host specificity so fundamental? Because it's, of course, it determines the parasite's niche, its persistence, and also its invasion potential, basically. So it determines how likely it is that parasites can actually shift to new hosts or to new environments and adapt to new hosts and environments. And in that sense, it's also important as kind of like as a risk of emerging infectious diseases. Like I don't know of any. I don't think we have to be afraid about chytrids like infecting people, but for example, for aquaculture industry, of course, it's important also to know like what is the risk that that these chytrids can enter the systems. And of course, it's a key factor that influences both ecological and evolutionary processes in host parasite interactions. So like host specificity has like, of course, it derives a lot of evolutionary processes. It determines kind of like parasite adaptation, host evolution, and co-evolution. And it also drives ecological processes like population dynamics and infection dynamics and community structure. So we have all this kind of eco-evolutionary feedbacks, like which host specificity is kind of a central trait to that. So that's why it's also kind of an interesting aspect to study. Then how do we measure it? Well, the very, very basic measure of host specificity is host range. And this is just, we can just count the number of host species a parasite can infect and completes its life cycle on or in. And with this host range or with this host specificity, we also commonly encounter the terminology of specialists and generalists. So specialists, parasites with a very high host specificity, so a narrow host range, and some authors define it specialist as really a single host parasite. And then on the other hand, we have generalists, you like determined parasites with a very low host specificity, a broad host range, and basically can be whatever, like multi-host, 2, 3, 4, 100. Right? So it's, you know, there is some space for variation in this trait. And just as a very, so with chytrids, we can basically expect everything is possible. So Alena already showed in her talk that chytrids, they are super diverse in their lifestyle and host use. And so I just plotted here, giving the relationship like facultative versus obligate and general specialist on like 2 axes. So facultative here meaning these are parasites that can both live without a host, so on organic substrates, and also use host as a parasite. So they have this both, they keep the both there, this lifestyle. And then obligate meaning that really they are only can live and can complete their life cycle on living host species. And so we can have, of course, facultative like generalists. Some examples here, and I put here DD, like the amphibian infecting chytrids, which is not phytoplankton hydrid, but I just put it here as a really extreme case that it can infect more than 1,000 amphibian species. But it's a facultative parasite. And then, but we also have, like, if we go to the obligate side, we can also have a generalist obligate. So meaning that this species, for example, it can infect several genera of green algae. So 20, 30 different green algae. Then on the opposite side, we have facultative parasites, which are reported at least as highly specialist. So they have a facultative lifestyle, but they really are like specific on one host species. And then on the other hand here, we have, of course, then the obligate ones and specialists, which we kind of intuitively expect that this obligate lifestyle evolves to increasing specificity with Astronella reflecting Hydroxyvo plictis, where we also have this host parasite genotype specificity in the system. And also like some chytrids are really specific on a particular cell within the host species. So already giving you kind of like the broad kind of all what is possible and what we can expect when working with chytrids and phytoplankton. But currently there is this kind of trend that the obligate parasitism leads to kind of more specialists and facultative are kind of more generalists. But we'll come that it's not so clear maybe. But then, of course, also multiple dimensions of host specificity. Like it's not only about host range, but we can also consider, like we can also include in this definition, like host preference. So it can be that a parasite has the same number of hosts species it uses, but it clearly has a higher prevalence in one of those. So it has a clear preference. So it's I would say it's still host specific in that sense because it has a clear preference. We can also include host phylogeny, so what is the diversity of host species a parasite can infect. Here again, it can be that they have the same amount of species, but in one parasite, the host species are very diverse, like divergent. Then it's kind of a low phylogenetic specificity, but it can be very high also when the host species are all very closely related. So this is also something that is can go in this definition of host specificity. And then another one that comes from Poulin's paper, the geographic specificity. Here again, it's more about parasite species. They can be quite specific at a local scale, but then in another environment, they might have a different host repertoire. So this is also an aspect that we can include when we think about host specificity. So it's not only about host range, but well, in the end, when we are talking about this or showing some examples, it seems that it's kind of difficult to surpass that simple host range measurement. Like it's challenging to kind of. And then a little bit about the niche perspective on host specificity, because we can like have this differentiation between the fundamental niche and the realized host range niche. So the fundamental host range would be all the host species that a parasite could use and infect under any kind of condition. But then the realized host range is really what we kind of observe and under a certain, like in a specific spatial temporal context. And of course, this comes very close also to like environmental filtering, what Alena also has already been explaining. So the fundamental host range can be much, much larger, of course, than what we actually observe in the field due to different environmental filtering factors like abiotic and biotic factors, which then usually gives a much smaller niche that we observe. And this becomes a bit like in the same, like as a classical niche theory, and it will determines also where we will find which kind of phytoplankton parasites pairs in nature. So, and of course here also migration, dispersal also plays here a role. Sometimes dispersal constraints which also prevent of having a global distribution. So in the end, what we observe is kind of like a small part of the potential. And now there are a lot of challenges, and this is now we are going to this rabbit hole. When I was preparing stuff like, well, maybe a question. I don't know if it's really the right moment, but I should have asked it earlier. But are Kite specialists or generalists? Well, we already know it because I showed it. I forgot to ask it before actually. So this is not working anymore. But anyway, forget about it. You know already. Yeah.
But maybe you can ask why are they specialists or generalists? Like what makes them? Other thoughts on that? Maybe you will tell more later.
Yeah, it's I think this is a discussion point. I don't have it specifically. I mean, we touched upon maybe a little bit, but there's not really a lot of empirical knowledge so far really on why. I mean, there are theoretical concepts that one can explore and test, but we will see about what are the challenges and limitations of getting to that data. And again, here there is, yeah, we are coming there, but so the first challenge, it's really trivial, but we have to identify. We need to identify phytoplankton hosting parasite species in a reliable way. And of course, we are working with protists, and now everybody knows now it's not so easy because they have a lot of like cryptic diversity, and before this kind of associations or how it was defined that type the host range of these cyclids, it was based on microscopy observations, most of this knowledge. And then we kind of realized that well, microscopic observations, and then what we can also use to measure or to assess this host specificity or host range. We can also use this laboratory cross-infection assays with culture isolates. So here we can kind of have an idea of a fundamental niche, and here with microscope observations like this realized niche. But again, these microscope observations, it really had like at some point it did not work because we have a lot of descriptive diversity, and that messes up everything. We have some examples. Here are 3 kitewit species infecting desmids in 3 different lakes. But like, does anyone dare to say if these are the same species or different species? So I always like say semi-cryptic because, of course, if one sometimes really clearly closely observes, then one can see maybe some kind of morphological differentiation in here, this trait of release of sporangia. But you really need to have that in culture, or like it's not something that you can just easily get from field observations. So no, these are all 3 different species and from different orders, Rhizophytialis, Chytrialis, so very, very divergent. And we also have that in the host. So that's another kind of problem, not only in the parasite, but also in the host. So these are 2 examples of, it's a colonial green algae, Pandorina, which is now Yammigisciella, and here Oidorina. So yes, they are different, but then they also can kind of be like this. So in this is the same, and then it looks kind of more like that, and here this one is like that. So there is this phenotypic overlap that also blurs if we can really morphologically distinguish them. And this was, of course, already known very, very long ago. People like, well, for those who can read French, it says here that it's really very difficult to distinguish these species. So that means that we need to have another way of like sequence those species, really distinguish them and from each other. And of course, this leads to also critical interactions. So in this example, we see that those 2 different genera they have a different set of parasites related to them. If we would have pulled them together, then we would not see this kind of different host specificity. But then we can also have the other way around. We can also have a species which infects different types of things, and it has a different morphology, but it is the same. So it's quite complicated in that sense. So it's not complicated, but we really need molecular tools. That's kind of like the key message here. We cannot do it based on morphology. The laboratory cross-infection assays, which culture isolates, has also inspired of challenges, of course, because, well, we can of course not test all possibilities in the lab. There are limitations to that, and what kind of hosts we test, what kind of environmental conditions we test, there are some limitations. Also, we often not really represent the real genetic variation in the population. So we work with a few strains, but we are not really reflecting maybe the whole potential of a population here. And then there's also cultivation bias. Of course, when we are cultivating this strain over 5-10 years on the same host, in the same conditions, we are doing experimental evolution. So we are also modifying maybe this traits. I don't want to make this a very pessimistic.
We assume you also come with a solution.
No, that's what I want to learn from you guys. So no solutions. It is discussion. And so the thing is that we are likely underestimating in many cases maybe the fundamental host range, or the potential of hybrids to be more generalist. And this is what I have experienced myself. So this is a paper that we wrote about this species description Taurastrum mices oculus, which infects a desmid species Taurastrum in Technin. And we kind of like already here, well, it suggests that it's specific, but we cannot exclude, just to be on the safe side. And luckily, because a few years later, just by accident, we kind of observed that it can also infect another genus, Taurodesmus, but only if we remove the mucilage layer of the host. So in this case, this was really very just, it was by accident that we kind of discovered this interaction. But then when we did experiments where we had treatments where we physically removed this mucilage layer from that host, we could clearly see that the prevalence of infection can get to the same amount than the original host. So it's only just this barrier. So, well, yeah, exactly. But also when we then had a little bit of a closer look, yeah.
You want questions in between, or so I'm just curious why you then still call it within the fundamental host range, because it can only be infected if you remove the mucus layer, right? That's what your Staurodesmus is only without this this envelope.
Yeah, yeah.
Is it actually susceptible?
Yeah. Maybe. So my idea was it's more kind of what here we underestimate the host range. It's kind of, or we underestimate the fundamental niche. I mean, because in this sense, how I see it, being the realized niche of these parasites, it depends in nature, like how this Taurodesmus, it depends on the conditions where it is.
Do you find it without the envelope?
Exactly. Yeah, we can find it, right? And I think I don't know if it's really very studied about like what are the conditions, why, like when it's really produced or not. But there are conditions that it might be there without, and in those cases, then these kind of interactions may happen. Yeah.
I just want to add that the scleroderma in the roots say is impacted in natural conditions, so this can happen.
Yeah.
I don't know if you observed, but I did. So it was impacted in the lake and the natural condition.
Okay.
So if this happened, so I mean, it's not only a kind of lab forcing removing the DNA. It can happen and it can be observed.
Yeah.
But then, sorry, did you then also observe that like a non-infected having mucilage as well, or was mucilage a trait that you saw in your system then as well, and was that associated with protection?
Well, when you observe under the microscope, it's not so easy to see this mucilage layer. So it's not 100% infected. I don't know if for me, mucilage can also be some strain specificity. So if there are sclerodermas that have it natural and sclerodermas that have it not, and this is the reason why some are infected and some are not.
Yeah, so we did not got any like into kind of like how like in dynamics in the field of this system. So it's kind of more a laboratory observation just to kind of indicate that when we would have used this host plane in the cross-infection experiments as it is, then we would not have like identified it as a potential host, but by modifying conditions, we can make this host range much larger. Like there's a larger potential. So this is a bit the message. But also when we went a little bit closer into like observing this interaction on Staurodesmus, then we saw that even the prevalence, when I asked them now about prevalence, the experiment was in that way that it was done with bull spores inoculated on host cells, and then after a very short time, fixing it and counting the attachment of these zoo spores. We also did another experiment just to show that it can also like reproduce. So that's all. But what we also saw that this Staurodesmus also had this resistance phenotype and in much higher numbers than the original host. So this kind of resistance phenotype can be also some kind of sign of local adaptation, because we were using this original Saurus from host species from clean, cultivated in the lab, and then exposed it to this Staurodesmus from another location. So they can be also this kind of effect of of local adaptation, that the parasite is much better at recognizing, or less good as in defending itself again compared to this allopatric species. But of course, it can also be a kind of a cultivation bias due to this long-term culture. Another story maybe to tell in this aspect is this species. It's Ghettoritzia hematococci. It's a parasite that infects Haematococcus. And it can also grow on PMDG medium. So this is an organic growth medium, which is often used for cultivating saprophytic hydrates. However, only without glucose. And I tell this story because when you see the authors, these are people that are specialists on saprophytic hydrates. So they have a lot of expertise on cultivating saprophytic hydrates. And they tested this and they found this out. But the future would have looked completely different if I would have found this hydrate. Because then I would say, I would test the NPTG medium and it would not grow. And I would say, this is an obligate parasite. This is just a stupid example, but it's just to show how different I don't dare to say anymore if something is host specific or has a larger host range. So, but luckily it's Joyce Longford that found this parasite. So we know that it also has a facultative lifestyle. Yes.
Yeah, inspired by that, do you have any hope that there would ever be like a general parasitism marker? Like a protein or anything that kind of would tell you this?
At the moment, I don't. At the moment, I don't think so. I don't know.
Can I just make
Yeah.
so this is my perspective.
Yeah.
I think it's dangerous to say it has a facultative lifestyle.
Mhm.
I think growing it on PMT plates, no G,
Yeah.
is a laboratory phenomenon. I think there
Yeah.
has to be and I get what you're saying completely, but there has to be a distinction between observing it in laboratory on an agar plate versus in nature.
Yeah.
Where I'm thinking of what you did with the chytrids that swap between pollen and that really is a facultative lifestyle, but it has a little asterisk on it for me. So just because it grows on an agar plate, I mean, there's lots of microbes that in nature only are parasites, but you can still grow them in the lab. Do you know what I mean?
Yeah. And you don't, okay, yeah, and but the realized lifestyle in the field, you would consider them not as like they are not growing on any kind of, that actually is, that's a nice comment. I mean, that's
like this is one of these raceway Hematococcus parasites, and I predict they never see it unless the Hematococcus is not there, you know, growing on the Hematococcus in nature.
Yeah, that's a good point. So yeah, this is a study about chytrids from the marine environment that was isolated, Mysis ariniensis. And here this is a kind of like an example of a chytrid with also a quite large host range, so it can infect multiple genera of dinoflagellates, but it has still a kind of like a relative preference for this Alexandrium genus. In this case, they also had two different strains, and you see also that strains can differ in their host range. So there are like this strain can, for example, grow on some procentrum, whereas the other not. So there is some strain variability, of course, also when assessing this host range and specificity. What you also see here are some kind of signs of local adaptation, like they used hosts of the Mediterranean Sea and hosts of other channel, like other locations. And here you also see some effect of this local adaptation. That's usually like the strains from the local environment. They are also kind of like the more they are more preferred than allopatric strains. But then again, so the same species, so this species was isolated for the first time from this coastal dinoflagellate bloom. Another study that identified or isolated this species from another different habitat type. So they were looking at macrophytes habitat type. And there they isolated also this species from. And they had several strains, and there they saw that one out of those strains could effectively infect pollen, whereas the others not. They could only infect, or they could only grow on dinoflagellates. So here also again this kind of idea of there is likely also high interspecific variation in species in their lifestyle or like host specificity. And here also again sampling may be biased habitat type. So we often, including like we are looking at a certain compartment of the ecosystem, pelagic, but then, well, not like there might be kind of the same species having different kind of lifestyles of interactions depending on the habitats. Yeah.
Is that a saprotrophic lifestyle on the left?
That's on the pollen, exactly. Yeah. So here it's infecting pollen. Yeah, and this is, well, the dinoflagellum is already so kind of far away that it's difficult maybe to see that it's still, but it was a dinoflagellum. So again, this kind of example of how an obligate parasite becomes, in this case, I would say a facultative parasite. And so what's happened or has been established now also in hybrid research is, I think this is has been a nice method development, is in situ single host hybrid pair isolations and for identifying host hybrid interactions in nature in the field. Because then we are not so bound on these cultivation biases, and so we can kind of pick what is really there, what has been established. Of course, it has also limitations and biases. So currently, it's still kind of low throughput. So we are manually picking it. So it likely just reflects the most dominant interactions and may underestimate less abundant ones, but it is at least also a method where what we can use to get more knowledge on the in situ occurrence of these host parasite interactions. So this was mainly established by Michael Degami and Christian Wolfsbacher. They have worked on this technique. So here's just the workflow. It's manually pick those cells, and then we can randomly amplify the DNA, like a whole genome amplification, and then we can do PCR and sequencing of the parasite, and ideally also of the host. And of course, nowadays we whole genome sequencing also becomes more and more possible to do that. Not only the 18S RNA, but one can also get whole genome information. So with this technique, this is a study of Michael from Lake Inba, and here with this technique they could clearly detect that it's not only a single parasite species that infects a host, but we often have a lot of very different, many different parasite species. So in this case, it was quite extreme. There were 8-9 different parasite species that were infecting the same host species, and they can also differ in their degree of host specificity. So it's not one, it's usually a lot of many parasites at the same time, or well.
Can I ask you something?
Yeah.
So how certain are they about the species distinction? Where do you draw a line that this is a species, or is it just a genotype?
I did not want to go in that.
Time for a break.
No, this is. I mean, the physical limitation. Is it what one can ask if it's actually? Is it still is it actually a real how can I'm more and more convinced that really delimiting species is always artificial. Of course, it is an artificial concept. So and there are, of course, with sequencing techniques, there are thresholds that people use, but then of course these thresholds can we need to also look at the biology then at the sites to see if those thresholds make sense, or if for some species there is a lower test and we see still some ecological difference, and then again, are these ecotypes or are they already? And I think in this sense, it's one can discuss a lot about what is a species and at what point they are really separated. But I think it's easier to see it as what is like the variation, and yeah, at some point, in the terms of host specificity, it might be then that this actually can be a kind of an indication. So some colleagues use host specificity to delimitate the parasite species. Then because if they can kind of, but then they can.
My question actually relates to the topic of your talk, which is about like the fundamental niche and the realized niche. All these concepts depend on like saying, okay, this species can use these species, and it's like where. It is.
Your artist is unified, right? So that's also, I mean, you can also look at interest-specific variation, depending on where you put the threshold.
Yes.
But the bottom line is there is some variation at some level wherever you put this threshold, right? That's your So you can also look at it as interest-specific, realized.
Exactly. How much time?
You still have like 18 minutes in total.
Okay, well.
But maybe 10 minutes.
Yeah. Then we go quite like maybe still this about so it is kind of information that we can like we have this single cell isolations. We got a lot more kind of information about parasite species to put in phylogenetic. We can look at phylogenetic relationships also of hybrids, and this is just to visualize like all the colored boxes with different colors. They are like these are parasite species, and the color is reflecting more kind of like the host taxa, so brown diatoms and greenish for green host species. But you kind of just see that parasites, it's not one phylogenetic group, so they are dispersed through the whole phylogenetic tree. So there have been independent evolution of parasitism or host specificity has been going on. And what you can also see, and this pattern results that parasites are often more closely related to saprophytes within their orders. And this kind of suggests that there likely have been quite a lot of frequent lifestyle shifts and host shifts that have created this kind of phylogenetic pattern. And what is like interesting, like maybe that there are some this order, the Zygoplictidalis order. This is currently an order where we only find at the moment obligate parasitic lineages, where you see like other orders, for example, Rhizopedialis is the extreme case where you have really a mix of different lifestyles. But this order seems to be some kind of example of a specialization of a parasitic lifestyle, and where maybe this life stage transitions might be not possible anymore. And when I just, and this is not this is just my kind of have been thinking about, like if there are maybe some kind of traits in hybrids that could indicate or could be indicated for what a specific lifestyle. And then these resting spores are kind of interesting in the sense that in Rhizopedialis, which is this kind of very more diverse group, the resting spore formation is occurring asexual. So that is the type of resting spore formation we see there. Whereas in Zygoplictidalis, which is this obligate parasite group, there the species have a sexual formation of resting spores. And it's kind of like interesting when thinking about co-evolution, host-parasite arms race, that the sexual reproduction might be some way of creating this genetic diversity and allowing this kind of co-evolutionary arms race, where you have that kind of trait not in that other lineage. Another kind of interesting, like just an observation, but I would think would be interesting to follow it further, like looking at these traits in Rhizophidialis, when the zoospores, when they don't find a host, and they can insist. So this is calcofluor white, so that means that they have built a chitin cell wall, and that is a kind of like a starting rhizoid. So they kind of, many species in this lineage, even if they don't find a host, they do insist, and they do have some kind of initial rhizoid formation in search for resources, but then don't find them here and die. Whereas here, I cannot like say exactly, but I never observed this kind of trait there. So just thinking about maybe there are also still some kind of morphological traits that could be indicated for different lifestyles.
Do you know how long can they stay in this form when they have this transistment, when they can survive without host?
Without before the existence. How long they can survive without that can depend on the environmental conditions. So I think temperature dependent quite a lot. So with higher temperatures, they usually have some higher metabolism and this faster, so they use up more energy. But yeah, a few days, I think maybe in the nature field, maybe up to week, 2 weeks.
The zoospore stage.
Yeah. Or maybe when it's kind of really cold environment, then it can last a bit longer, actually saving energy. I don't know if they can kind of decide to go immortal and then wait.
Related to that, I was wondering, did you come across anything that could indicate a switching maybe, so that you have a parasitic lifestyle, but then they actually use a saprotropic lifestyle to survive longer? Well, survive, I mean, then basically they become saprotrophic, reproduce, and then they continue infection again as soon as the host is around. Is that the whole point of being saprotrophic? Did you come across any of that you kind of have this?
I don't know.
No.
I don't know. I think that's the platform to like this mechanistic basis of whole specificity, and I think we are like not
in the saprotrophic ones in BD, the frog one. Frog amphibian mucin
exactly
initiates the transition.
Yeah.
And then we've seen in Arglobosum, which is a saprotroph, which is a chitinophilic saprotroph, that chitin initiates transition.
Yeah.
So it could be that if you gave them some host exudates and some algal exudates, that might make them switch on. It might be something to try experimentally.
Yeah. Now I think with this kind of experimental work, like there's a lot of things that we can test. And I think that's why I said I'm not working actually on host specificity again, because I don't all for you here. I only show some things what we like. Maybe very briefly about like because are there costs for example, because why are not all species and generalists? It would make it easier, of course, if you keep that feature always there present, and you just be a specialist and when your preferred host is there. But so are there costs for being a generalist? And there have been some studies that have kind of indicated that yes, there are these costs in this system again with deomyces. So the one where we had a strain that was this generalist that could infect both pollen and dinoflagellates, and those strains that only can infect dinoflagellates, they see that the strain with the generalist strain infects much less. So here, this actually shows you the curve of the host abundance. So they did a cross-infection experiment and looked at the infection rates of the chytrids. And this strain is this pollen strain. and the host abundance is actually equal to the one of the control. So it cannot, it could not control, in this case, this host population. So whereas the other obligate strains, they could kind of like induce a high prevalence and control. So there is, in this case, it shows that there might be some fitness costs related to this more generalist lifestyle. Also another study by Michael and Kensuke, they also could show, so they did a bit of a similar experiment looking at growth rates of generalist and specialist strains, and clearly also found that specialist strains tend to have higher growth rates than these more generalist strains. There seems to be some cost related to that. So very briefly, I think I will not be able to present everything, but just to show like what we kind of like try to do and to look with this combination, like methods to look at temporal and spatial dynamics of these host hybrid parasite interactions, and identifying maybe like these specialists and these different lifestyles and how they are, how they co-occur, or what kind of like the dynamics of these different lifestyles. And in this case, I think, like, of course, high throughput sequencing has been a game changer to kind of be able to have this temporal and spatial resolution of community diversity. But then, of course, we still have this problem that we cannot infer from those host parasite interactions. But by then combining these different methods, we might have some kind of better knowledge of, like, we can disentangle maybe like specific species interaction. So this is what I kind of have to do in my research, to combine this eDNA approach, then at the same time using microscopic observations to quantify infection, using targeted isolation approaches, the ones that I kind of explained before, where we sequence, so we can do cross-infection assays of cultures if we obtain those to infer some of those traits of lifestyle and host range, and then we can create this kind of local reference sequence base for this local environment, and matching those then through the with the eDNA community, where we then can kind of like pinpoint, okay, this is so it's, of course, I explained already all the limitations that are there, but it's a way now of like what we have been doing to at least have a bit of a better idea of specific host parasite species dynamics. And this is just from Lake Stechlin, an example where we made this using this combined isolation cross-infection approach, this host hybrid interaction matrix, where we already found like the results that I already showed before, that we have multiple hybrid species that are associated with a single host species. So we often have multiple ones. Many of those, based on our methodology and our limitations, like I always be hesitant, but many of those with this deterministic approaches were defined as specialists, and we also have a few generalists and few facultative parasites. So in this system, we also find all these different interactions and lifestyles back again. And what we can then, again, by saying when we combine this eDNA approach, then we can kind of map the dynamics of different species. And here we then find that during, for example, a Fragillaria bloom in spring, we can have this kind of co-occurrence of specialist hybrids and more generalist hybrids. So we see that kind of like that is possible. And then thinking about like cost of generalists, what's like, because one can think, okay, why, like if there is a specialist, it could probably easily outcompete a generalist. And then it was very interesting that we, at the same time, we observed during that period an hyperparasite interaction on one of the hybrids. Unfortunately, we cannot relate it to which one. This is morphology based. So, however, when we correlated the microscopic prevalence of these hyperparasites with the ASV of the specialists and the most abundant Rosella mycota ASV, which are often hyperparasites, they kind of corresponded. So this was kind of interesting. Maybe this hyperparasitism on the specialist strain might be some kind of mechanisms where we can also have this co-occurrence and of both specialists and generalists at the same time. Other dynamics, for example, on Astrionella. So here we identify 2 parasite species. Wrapping up? Okay, so I will tell this story and then. And yeah, so this were identified as 2 specialist species based on our cross-infection assays, single cell isolation. And here we have a little bit different dynamics. So here they are not co-occurring, but they have kind of different niches. So Zygoplictidalis has these summer peaks, which correspond to high, like this is periods of infection, so they have these massive epidemics, whereas this other one is kind of there when Astrionella is in low abundance, and even when we don't see actually any kind of infections there. So this also again reflects that maybe this Rhizophidialis, maybe it has a kind of like a broader host range. And this is also something that we can maybe try to see, like identify, okay, relate infection patterns and occurrence patterns with specialist or generalist lifestyle. So the ones that are really the specialists, and they have this kind of massive epidemics, they usually have this kind of very like, short-term peaks, but then maybe this kind of more flattened out and continuous occurrences, and sometimes some kind of flash. This kind of might be also some kind of indication of a more generalist or a broader lifestyle. And okay, I don't going to talk about the Baltic then. We are going to talk about this.
So maybe this is all extra for, you know,
yeah,
anyone.
I'm sorry.
No, we have questions as well, so we could do.
I said it was a rabbit hole, right? I did not lie. I want like maybe just myself. I kind of like think that this whole specificity, like it is likely common in hybrids. In terms of host preference and that we have due to local adaptation, we kind of have this more specialized interactions going on, but there is the potential is higher, and I think this is still interesting that to realize that that there is this because that means that there might be actually more evolutionary flexibility that prevents this hybridism from really becoming so specialized that we cannot switch anymore, or we cannot adapt anymore to any new environments. And just as an implication, or in ParAqua, also interested in like applications for aquaculture. I think this is something to keep in mind. That yeah, there might be some risks. So key takeaway, I think, is still, yeah, that's again what I said. So it makes these host parasite interactions much more dynamic, I think, than we currently still assume. And yeah, how to go further. I have no this is, I'm here to learn from you guys of what we actually like. what we could maybe do to overcome this kind of limitations also of identifying this species, these interactions, and also coming over this fact of that we cannot test everything, or like to come to some kind of way of dealing, like to also tackle this also a bit more testing theories, right? Like now we are I have showed a lot of observations, we are observing, but where do we go from observation to really hypothesis testing and predicting? And I think this is one of the things is which could help is to also get a better understanding of the mechanistic basis of specificity. This is something that we currently are missing. And with
Thanks.
yeah.